Hello, and welcome to part 2 of our adaptive immunity lecture series. And here we're going to get into the whole nitty-gritty of adaptive immunity and how it kind of starts to differ from innate immunity. Remember, innate immunity, we were born with it. Adaptive immunity, we have to encounter the pathogen before it can actually exhibit a response. Okay? And so if you're ever thinking about reviewing adaptive immunity, I would come back to this slide. This slide kind of spells out all of adaptive immunity and how it takes place in our fluids, how it takes place in our nucleated cells, and how it takes place in our macrophages. Okay? And remember, macrophages and nucleated cells are going to be dependent upon the major histocompatibility complex. Okay? And there are two different types, and we'll get into those. But basically, we have humoral immunity and the fluids. Okay, so how do your fluids regulate pathogens? Okay, your blood, your lymph, your CSF, every kind of thing. If there is a pathogen encountered, it's going to be a B cell. Okay, and this B cell we'll get into, but they are matured in the bone marrow, hence the B. Okay, and then when they encounter a pathogen, they will activate and will become a plasma cell, and then they release antibodies. Okay, and then antibodies are free floating in the blood and in the serum and in our fluids. Okay, that's why we call it humoral defense, because the ancient Greeks called the four humors the fluids of the body. Okay, so that's where we get it from. But you can kind of relate it to fluids, and okay, these would be patrolling our body because they are being circled by the heart. Okay, and so the antibodies are in our blood, and the antibodies we'll see are like the quintessential sign of adaptive immunity. Okay, and what we think of when we think of adaptive immunity. Okay, nucleated cells have their own version of saying that they're sick. Okay, so any cells that we have that are infected will exhibit the signs that they're infected and present an antigen on its major histocompatibility complex, and that is found and read by a cytotoxic T cell. Okay, and they activate, and when we say activate, we kind of mean proliferation. Okay, proliferation of these T cells will then target these pathogens and these nucleated cells that are infected with cytolysis, okay, and destroy the cells that are infected before they can spread some more. Okay, and again, what we mean by proliferate is the T cells undergo lots and lots of mitosis. Okay, and we learned about macrophages and how monocytes become macrophages, and macrophages are found in a lot of our different types of tissues, liver, brain, and they're called different types of cells based on where they're found. And so they find the pathogen, phagocytize it, engulf it, and then they'll present the signs of this to a helper T cell. Okay, and remember, with T cells, both the cytotoxic and the helper, they're both matured in the thymus. Okay, so bone marrow B, thymus for T. Okay, then they activate, meaning they're going to release cytokines. Okay, and those cytokines are going to tell other cells to come, and then they will destroy the infection and proliferate through mitosis. Okay, so again, this is a great overall review slide, and it shows just how we're going to take care of all the infections in our cells. And note, we don't see red blood cells on here. Okay, red blood cells are not nucleated. Okay, so if your red blood cells get infected, they end up either getting destroyed by the macrophages, um, uh, and that's just over time, or they um, just wait until they're destroyed naturally over their life cycle. Okay, and so it's important to note that the red blood cells do not have a major histocompatibility complex. Okay, so in continuing with innate immunities, first and second line of defense, adaptive immunity also has the term third line of defense. Okay, and we can see that the bone marrow and the thymus are integral parts of the third line of defense. Okay, because these are where the sites of B and T cell production and T cell maturity. Okay, and then so they can learn to distinguish self from non-self in the thymus. Okay. The spleen filters blood, trapping foreign particles, presenting them to the lymphocytes. And the same thing about the lymph nodes. And we learned about this in the first couple slides of innate immunity, that the spleen and the lymph nodes act kind of like a netting, and they kind of trap the pathogens there before they can spread and give us an infection. Okay, so the third line of defense is the adaptive immunity, and it also includes leukocytes. Okay, and when we learn about the immune response, we learned about immunity, we've learned about immune cells. Now we're going to learn about the immune response. Okay, and remember how we said that secondary adaptive immunity is slower and it takes time, but after that it's more targeted and specific and ruthless. Well, that's kind of what this whole graph kind of says. It says when you first encounter it, you rely solely, mostly on your innate defenses, okay? And the innate defenses kind of hold it back. And then when you finally get sick, the leukocytes, the natural killer cells, try to destroy it, but they do so slowly, okay? And then eventually your adaptive immunity kicks in and destroys the pathogen, and you get better. And then, obviously, or you don't. Okay? But then again, when you see this same pathogen again, all right, your next immune response is going to be greater. Okay? You're going to have a way more abundant immune response. Okay? And how do we grade an immune response? We grade it with the detection of antibodies, which we said were the quintessential adaptive immunity trait from the previous slide, right? So in order to even test for our immunity, we look at the initial antibodies, okay? So we need adaptive immunity to even kind of tell us how much immunity is going on, okay? Notice the secondary response is faster and provides a much higher concentration of the antibody, okay? So now we need to kind of discuss what antibodies are, okay? But in order to know what an antibody is, we have to know what an antigen is, okay? So an antigen is any molecule or macromolecule that reacts with the components of our immune system. So at some point in history, you have learned that this molecule is reactive, okay? And in that sense, it reacts with something in your immune system. It tells it to react. That is an antigen, okay? If it doesn't react with you, okay, then it doesn't elicit a response. It's not an antigen. You don't really know about it, okay? But an antigen may contain several motifs, okay? So what do we mean by motif? It just means like a recurring pattern. So we mean like a pattern. Here is a pattern that is recognized by a certain antibody. Here is a pattern that is recognized by a certain antibody. And here is a pattern that is recognized by a certain antibody. Okay? And, with anything in biology, if you see any kind of like shapes like that, that's kind of just like the fuzzy ball reading that they get when they look at it under an electron microscope or a NMR reading. They kind of get these crystal structures, and this would be kind of like what they see. Okay, so these different motifs, these different types of antigen that are recognized are called epitopes. Okay, so an antigen can have multiple epitopes, and each one can react differently to you. A certain epitope might not elicit the response that another epitope would. Okay, and all of it, anything that reacts with your immune system, would be the antigen. Okay, so now we see what an antigen is, and we see what an epitope is, and we see the inverse of the epitopes, and that inverse of the epitopes is what we see here in the antibodies. See how they're all kind of that shape that I was drawing? Well, a typical antigen with multiple epitopes can have different antibodies that bind to different epitopes. Okay, so different antibodies for different epitopes, even though it could be the same antigen, like pollen or dander. Okay. All right. So when we look at the antibodies that are around our antigens and they bind to it, we can, like I said, crystallize this structure, and we can see that it kind of creates this Y shape. Two halves of a Y come together, and they get it from this. Okay, so they see what it looks like here, and they kind of draw it for ease over here. Okay, and now we can kind of look at the regions here. Okay, so the heavy chain area, the FC region, okay, is the part that binds to one of our cells. Okay, so our cells, and the Fab region is very specific, very specific. Okay, they can only recognize certain epitopes of certain antibodies, maybe only one, or of antigens, maybe only one. Okay, so this Fab region, the Y part, or the V part, if you will, is the variable part that recognizes the parts of the pathogen. Okay, and that this isn't big enough for the whole entire pathogen, right? It only needs to detect a piece of it. Okay, and so we'll end with the 5 classes of antibodies, okay, or immunoglobin classes. Okay, so certain antibodies are in the immunoglobin class. Okay, and these immunoglobins have certain roles. Okay, and remember, we have gamed, and we can remember gamed to remember our 5 immunoglobin classes. Okay, and we can already look at D. Okay, it is less than 1% and is mostly negligible. Okay, we can also see that allergy is for IgE. Okay, so allergy for IgE. Okay, so those are the 2 easier ones. All right, and then we got to learn IgG is the heavy hitter. Okay, IgG is the main player, 80%. Okay, So always trust your gut, and that it's probably IgG. Okay, it's going to be more numerous and more responsive. Okay, there's a bunch of IgGs floating around, and that's because they can elicit all of these types of functions. Okay, so very important. The IgM, on the other hand, is not as abundant, but can bind to 5 different antigens at once. Okay, and they can also elicit a whole bunch of responses. Okay, and they are really a good player in um, secondary immunity. Okay, and then IgA is notable because those are in our tears, saliva, okay, and mucus. All right, and so that's where once it traps the pathogen in that mucousy or saliva matrix, we have some antibodies for it, and that's in the presence of IgA. Okay, and those are secreted usually, so they have that secretory component. Alright, so in the next videos we'll get to go see what each of these functions are that the Ig antibodies actually can elicit. All right?